Hello everyone. Today the topic of discussion is evolution of sporophytes and bryophytes. This is both for UPSC students of botany and also for botany honors students. In UPSC there are questions for long answers and also for short answers. In the latter case there is a word limitation. For long answers the discussion should be addressed in detail. An indication for short answers is also given here. For helping UPSC students who has come from non-biology or non-botany streams, a recapitulation or introduction to the subject is given. Evolution of sporophytes and bryophytes. Recapitulation. Haploid refers to the presence of a single complete set N of chromosomes in an organism cell. Diploid is a term that refers to the presence of two complete sets 2n of chromosomes in an organism cell, with each parent contributing a set of chromosomes to the pair. Gametophyte. Phyte is plant and gametophyte is a plant that produces gametes. The haploid N phase in the life cycle of a plant is known as the gametophytic phase or gametophytic generation. In alternation of generation in a life cycle of a plant, gametophyte develops from the spore N and produces gametes or sex cells by mitotic cell division or equational cell division. These sex cells may be referred to as gametes. Male sperm and female eggs housed in male and female gametangia called antheridium and archegonium respectively. This is followed by the process of fertilization, that is, union of male and female gametes producing a diploid zygote 2n. Now sporophyte. A diploid 2n phase in the life cycle of a plant is known as the sporophytic phase or sporophytic generation. In alternation of generation in a life cycle, sporophyte develops from the zygote 2n and produces spores by meiosis or reductional cell division. The spores then develop into a haploid N gametophyte. Now bryophyte. The main plant body of a bryophyte is a gametophyte. That is, the gametophytes are dominant, independent, longer lived, and constitute the main plant body. The diploid sporophytes are comparatively rudimentary structures and remain attached to and are nutritionally dependent on the gametophyte. In the evolutionary scale starting from Bryophyta has a gametophytic plant body. The next higher scale is the Pteridophyta, which has a sporophytic plant body. Next is the Gymnosperm, which also has a sporophytic plant body. And lastly, the highest one in the evolutionary scale is the Angiosperm, which also has a sporophytic plant body. So it is obvious that sporophytic plant body is more advanced. Now, Bryophyta consists of three main groups: Hepaticopsida, Anthoceratopsida, and Bryopsida. Now, the relevant points of Hepaticopsida. This is the simplest of the three. They are also called liverworts. Here, the gametophytes are either dorsiventrally differentiated simple thallus, examples Sphaerocarpus, Rixia, Barkensia, Pelia, or they are radial and erect, having leaf-like appendages, that is folios, example Porella of the Jungle Manualis. The sex organs are born either from superficial dorsal cells or terminally. Sporophyte are either simple or differentiated variedly into foot, seta, and capsule. Columella absent. Sporogenous cells are of endogenous origin. Classification is as per ICBN, that is, International Code of Botanical Nomenclature. The second is the Anthoceratopsida, also called hornworts. Gametophytes are dorsiventrally differentiated, simple. Internal tissue homogeneous. Sex organs are embedded in gametophyte. Sporophyte is differentiated into fruit and capsule. The interesting features are the presence of meristematic tissue. Lower part of the capsule remain meristematic. This helps in unlimited growth of the sporophyte. Second is the presence of columella. The internal or central cells form a cylindrical structure named columella, which gives support and aids in conduction. Generally, sporogenous cells are of amphithecial origin. Example, Anthoceras. Third is the bryopsida, also referred to as mosses. Here, mature gametophytes possess erect, usually radially symmetrical shoot or stem-like structures called corollas, with leaf-like appendages called phyllodes. Sex organs develop from superficial cells at the top of the apical or axillary meristeme. Sporophytes are 1. Usually differentiated into fruit, seta, and capsule. 2. Usually of limited growth, however, many mosses have long continued growth. 3. Presence of columella. 4. Presence of chlorophyllous cells and stomata in the apophysis region. 5. Presence of structure of spore dispersal called peristome. And 6. Sporogenous cells are endothecial or amphithecial in origin. Example, Pogonatum, Polytrichum, Funaria, etc. So you can see a gradual specialization of the sporophyte and bryophytes in the order Herpetocopsida simple to Anthoceratopsida to Bryopsida most specialized. Though we tend to view a gradual upward evolutionary trend within the bryophytes from Herpetocopsida to Bryopsida, there are actually two opposing views. Now we come to the answer proper. Answer: evolution of sporophytes in bryophytes. There are two opposing theories regarding the evolution of sporophytes in bryophytes. 1. The theory of progressive evolution, and 2. The theory of regressive evolution, that is, the evolution of sporophytes due to progressive reduction or simplification. Now first we deal with theory of progressive evolution, that is, the evolution of sporophytes by the progressive sterilization of potentially sporogenous tissue. Now, who supported the theory? This theory was advocated by Bauer, 1908 to 35, and supported by Cavers and Campbell. According to this theory, the primitive sporophyte of bryophytes was simple and most of the sporogenous tissue was fertile. Example, Rixia. And from such a sporophyte, the more complex sporophytes, examples mosses, have evolved by the progressive sterilization of potential sporogenous tissues. This theory is also known as theory of sterilization. The sporophytic phase begins with the formation of a diploid zygote within the venter of the archegonium. Now, progressive evolution or progressive sterilization occurs in a number of stages, 8 stages in total. First stage, the simple sporophyte of Rixia. Here, the zygote has no polarity. It divides by several divisions and differentiates into a single layered outer sterile jacket from the amphithecium and inner multicellular mass of sporogenous cells from the endothecium. Now, you must be wondering, what is amphithecium and what is endothecium? Amphithecium is the outer layer of the sporophyte and endothecium is the central cell mass of young sporophyte. The endothecium differentiates into fertile spores and very few sterile nerve cells. Nerve cells are absorptive, nutritive cells. So sterile 2N cells consist of 1. Single layered jacket and 2. Few nerve cells. The nerve cells later perishes. The fertile cells give rise to spores. So in a mature sporophyte of Rixia, sterile cells consist of only a single layered jacket. Now we come to the figures. Diagram shows rickshaw sporophyte having only a capsule. Here in the diagram, the capsule shows a single layered sterile jacket on the outer side and spores tetrad inside. It also shows disintegrating nerve cells. So mature sporophyte consists of only capsule having a sterile single layered jacket on the outside and the spores inside. Now the second stage. The second stage is exemplified by Corsainia. Development of sporophyte. Zygote transversely divides into hypobasal cell and epibasal cell. Hypobasal cell give rise to small sterile foot. Epibasal cell give rise to amphithecium on the outer side and endothecium on the inner side. Amphithecium give rise to sterile single layered jacket of the capsule. Endothecium give rise to fertile spores and sterile elongated nerve cells. So sterile 2N cell consists of foot, single layered jacket, long elater-like nerve cells, and fertile cells give rise to spores. So foot is the new addition of the sterile tissue, and elater-like nerve cells here remain also in the mature state. Now the diagram of Corsainia. The sporophyte in Corsainia consists of capsule and a small foot. Capsule has a single layered jacket on the outer side. Within the capsule are spore tetrads and nerve cells. Each spore tetrad gives rise to 4 spores. Here we come to the third stage. The third stage is exemplified by Spherocarpus sporophyte. It consists of a sterile bulbous foot, a narrow sterile seta and capsule. Sterile bulbous foot and short narrow sterile seta are both developed from hypobasal cell. Outer single layered sterile jacket of capsule is developed from amphithecium, and sterile nerve cells are developed from endothecium. So, sterile 2N cells consist of 1. Sterile bulbous foot, 2. Short narrow sterile seta, 3. Sterile jacket, and 4. Sterile nerve cells. Fertile cells give rise to spores which are developed from endothecium. So in the third stage, sterile foot becomes bulbous and sterile seta is added. Diagram of Spherocarpus. The sporophyte of Spherocarpus consists from top downwards, 1 capsule, 2 small seta and 3 bulbous foot. The capsule has an outer single layered jacket. Within the capsule are the nerve cells and spore tetrads. Fourth stage. The next stage can be found in Targonia, where the sporophyte consists of a sterile bulbous foot from hypobasal cell, a sterile narrow but longer seta from middle cell, and a capsule from epibasal cell. Epibasal cell give rise to amphithecium and endothecium. Amphithecium give rise to sterile one-layered jacket. The important point here is that about half of the endothelial cells produce fertile sporogenous tissue, while the remaining half give rise to sterile elaters with 2 to 3 spiral thickening. Hence, in Targonia, more sterilization of sporogenous tissue is done in this way. So, sterile 2N cell consists of 1 bulbous foot, 2 narrow but longer seta, 3 outer single layered jacket, and 4 half of the inner endothelial cell, giving rise to sterile elaters with spiral thickenings. Remaining half of the endothelial cells are fertile, giving rise to spores. Diagram of Targonia sporophyte, longitudinal section. It consists of, from top to bottom, 1 capsule, 2 comparatively well-developed seta, 3 bulbous foot. The capsule has a single layered jacket on the outer side and elaters with thickening and spore tetrads within. Since half of the endothelial cells give rise to elaters, you can add 3 more elaters in the diagram. Diagram of Targonia sporophyte, longitudinal section. It consists of, from top to bottom, one capsule, two comparatively well-developed seta, three bulbous foot. The capsule has a single-layered jacket on the outer side and elaters with thickening and spore tetras within. Since half of the endothelial cells give rise to elaters, you can add 3 more elaters in the diagram. Now the fifth stage. This stage is found in Markensia. Here further sterilization of sporogenous tissue has occurred. Development of sporophyte. Hypobasal cell give rise to sterile foot and portion of sterile seta. Epibasal cell give rise to remaining portion of the sterile seta and capsule. Capsule consists of sterile unilayered jacket, sterile elaters and spores. Hence, sterile 2N cells of the sporophyte consists of 1 sterile broad foot, 2 sterile mesopseta and 3 capsule. Capsule consists of a single-layered sterile jacket and large number of sterile log elaters with spiral thickening. Fertile cells give rise to spores. Diagram of the sporophyte of Markensia, longitudinal section. The left-hand diagram shows the longitudinal section of the sporophyte in the normal form. The right figure is a diagrammatic and simpler representation where parenchymatous cells are not drawn. Now, the labelings from the top to bottom. 1 broad foot, 2 mesopseta, 3 capsule. Capsule is with a single layered jacket on the outer side and long elaters with spiral thickening and spores within. You must be surprised as to why the sporophyte shown is inverted, foot at the top and capsule at the bottom. This is because the sporophyte hangs upside down from the archegonial stalk of the gametophyte. Now we come to the sixth stage. This stage is represented by some members of Jungermanialis like Pelia, etc. Here, more sterilization of sporogenous tissue has been observed. Now how? Here sporophyte is differentiated into foot, seta and capsule as usual. But the capsule have bi-layered jacket, so addition of one extra layer to the jacket. The sporogenous tissue produce along with spores mass of sterile fixed elatophores at the bottom of the capsule and diffuse elaters. So elatophores are new addition. Hence sterile cells are foot, long seta, bilayered jacket of the capsule, bunch of fixed elatophores and elaters. Fertile cells are spores. Figure shows the diagrammatic representation of sporophyte. Parenchymatous cells are not drawn here. Sporophyte consists of sterile foot, long sterile seta and capsule. The capsule consists of bilayered sterile jacket or capsule wall, 2 bunch of sterile elatophores fixed at the bottom of the capsule and elaters. Fertile cells are the spores. It must be noted here that bilayered jacket should be indicated in the drawing or in the labeling, or both. Seventh stage. Seventh stage is illustrated by members of Anthoceratopsida like Anthocerus. Here marked differentiation in the tissues has been noted. Outer 4 to 6 layered sterile capsule wall and fertile sporogenous cells are derived from amphithecium. Capsule wall differentiates into epidermis with stomata and chlorophyllous cells. This makes it partially independent with some assimilatory power. The centric columella is derived from endothecium. It is composed of vertical rows of sterile cells. Function is conduction and support. At the base is the meristematic tissue which enables continuous growth. Sterile 3 to 4 celled long, simple or branch pseudoelaters are derived from sporogenous tissue. Fertile spores form from sporogenous cells. Summary. 7th stage. Example Anthoceros of the Anthocerotopsida. Labelings of Anthoceros sporophyte shown previously from top to bottom. 1 spore, 2 pseudoelater, 3 spore thread, 4 jacket or capsule wall, 4 to 6 layered, 5 columellar, 6 spore mother cell, 7 archosporium, 8 stomata, 9 meristematic zone, and 10 foot. 8th or final stage. Summary. The members of Bryopsida, like Funaria, Polytrichum, Pogonatum, etc., show the highest degree of sterilization. The sporophyte is differentiated into a sterile foot, a long sterile seta, and a capsule. The sterile tissue of the capsule is highly differentiated and comparatively advanced. It consists of 1. Operculum, that is the lid, 2. Peristome, a structure which aids in spore dispersal, 3. Tympanum, a drum-like structure just behind the peristome, 4. Multilayered jacket or capsule wall, 5. Air cavities, 6. Trabeculae, chlorophylous tissue in air cavities, 7. Columellar, this is the tissue for conduction and support, and 8. Apophysis, chlorophylous tissue, this is for photosynthesis and also for conduction. The sporogenous tissue is restricted to the spore sacs only, hence it forms a negligible portion in the sporophyte. Hence it is apparent that along with progressive sterilization of sporogenous cells and increase in mass and length of sporophyte, there is also differentiation of cellular mass into tissues, leading to partial independence and the tendency towards land habit. Diagram of sporophyte of Pogonatum. The diagram shows from top to bottom. Top, capsule. Next is the apophysis, that is the chlorophylous tissue having stomata, and at the bottom is the seta. Now the capsule consists of, from top to bottom, 1. Operculum, that is the lid, 2. The peristome, that is the specialized structure for spore dispersal, 3. Tympanum, a drum-like structure just beneath the peristome. After that, on both sides, from outer to the inner side, are 1. Multilayered capsule wall, 2. Outer air cavity having inside trabeculae, that is the chlorophylous ribbon-like tissue, followed by internally spore sac containing spores, and interior to this is the inner air cavity with trabeculae. After that is in the central area, that is the centrally placed, is the multilayered columellar for conduction and support. Summary. Theory of progressive sterilization was advocated by Bauer 1908 to 35 and supported by Carroll 1910 and Campbell 1940. According to this theory, the primitive sporophyte of bryophytes was simple and most of the sporogenous tissue was fertile. Example, Rixia. And from such a sporophyte, the more complex sporophytes, example mosses, have evolved by the progressive sterilization of potential sporogenous tissue as follows. First stage, example Rixia. Here, sterile, that is 2N, cells consist of 1. Single layered jacket, 2. Few nurse cells, but these nurse cells later perishes. so there's only single layered jacket. Second stage is Gaussinia. Here sterile 2N cells consist of 1. Single layered jacket and 2. Persisting long elater-like nerve cells and 3. Small foot. Third stage, example Spherocarpus, here foot is bulbous, and 4. This addition of seta. Fourth stage, example Targionia. Here there is 1 sterile bulbous foot is larger, sterile seta is narrow but longer, and 3. Half of the inner endothelial cells give rise to sterile elaters with spiral thickenings. And other half is the sporogenous cell. So this point, the half of the inner endothelial cells give rise to sterile elaters with spiral thickening is important. Now the fifth stage, example Markensia. The sterilized portion of the sporophyte consists of 1. Larger foot. Larger is important. 2. Massive seta. Massive is important. 3. Single layered jacket of the capsule, and 4. Large number of sterile elaters, sterile long elaters with spiral thickening. So here the elaters are long with spiral thickening. Now sixth stage, example is the Palia. Here the sterilized cells of the sporophyte consist of 1 foot, 2 long seta. Here in the figure you can see long seta. 3. The important thing is that bilayered jacket of the capsule. Here the first time there is the bilayered jacket. 4. Bunch of fixed elatophores. So elatophores is also new here. And 5 is the elaters. Seventh stage, example Anthoceras. Here further sterilization of sporogenous tissue has been observed in the formation of 1. Multi-layered capsule wall with stomata. Now here's the first time we have multi-layered capsule. In Palia there was bilayered. Now 2 is the multi-layered columella for conduction. So columella is also new here. 3. Sterile 3 to 4 cell pseudoelaters without thickening band and meristematic tissue for unlimited growth in addition to foot. Here you find the figure, meristematic tissue is just above the foot. Now next is the 8th stage, example members of Bryopsida like Pogonatum. Here's the sporophyte of Pogonatum. The sterile tissue of the capsule is highly differentiated and comparatively advanced. It is very different from the others. It consists of 1. From the top, operculum, that is the lid. 2. Peristome, structure which aids in spore dispersal. 3. Tympanum, drum-like structure just beneath the peristome. Then on the outer side, many-layered jacket or capsule wall. From the jacket inwards are outer air cavity with trabeculae, that is the chlorophyllous tissue within the outer air cavity. Then the inner ear cavity, inner to outer ear cavity. Inner ear cavity is also with trabeculae. In the center is the columella. Columella is for conduction and support. At the bottom of this is the apophysis, which consists of chlorophyllous tissue for photosynthesis and stomata. And lastly at the bottom is the seta. So you have read the theory of progressive evolution of sporophyte and bryophyte, which is progressive sterilization of potentially sporogenous cells together with differentiation of the sterilized tissues. Now we come to the second theory, that is the theory of regressive or retrogressive evolution or evolution of sporophytes due to the progressive reduction and simplification of sterilized tissues of the sporophyte. This theory is supported by Church, Kashyap, Gober and Evans. According to this theory, Rickshaw sporophyte of the Hepaticopsida, which is essentially a capsule containing a single-layered jacket and a mass of sporogenous cells and a few non-permanent sterile nacelles, is the most evolved. The sporophytes of Bryopsida, on the other hand, showing the highest degree of sterilization and specialization of the sporophyte and possessing comparatively less sporogenous tissue, is the most primitive. According to this theory, from Bryopsida, there is a gradual reduction of sterilized cells and simplification of tissues. It encompasses 1. Reduction in the sterile tissues of the capsule with simultaneous increase in the amount of sporogenous tissues. 2. Simplification of tissues and loss of specialized tissue structures. Loss of specialized tissue structures include 1. Loss of spore dispersal mechanism, peristome, present in Bryopsida only. 2. Gradual loss of photosynthetic tissue, which includes chlorophyllous cells, thereby restricting the partial independence of the sporophyte. 3. Gradual loss of stomata. In Funaria, polytrichum and sphagnum, stomata are present in apophysis region, but in sphagnum the stomata are rudimentary and non-functional, while in Hepaticopsida stomata are completely absent in sporophytes. Next, loss of conductive tissue. Conductive tissue is present in members of Bryopsida and Anthoceratopsida, but it is absent in Hepaticopsida. Next, loss of meristematic tissue. Meristemic tissue is present in Bryopsida apophysis region and Anthoceras, but it is absent in others. Multilayered capsule is found in most of Bryopsida, Anthoceratopsida, and Junga Manualis. It got reduced to single-layered in Markinchialis, Markinchia, and Rickshaw. Next, gradual loss of foot and seta. Foot and seta, which are well developed in Bryopsida and some Hepaticopsida like Palea, Markinchia, etc., are much reduced in the sporophyte of Targonia of the Hepaticopsida, while in Corsinia of the Hepaticopsida, seta is absent. In Anthoceras, the sporophyte is made up of a foot and an elongated capsule only. Seta is here absent also. Finally, in Rickshaw, foot and seta are absent and the sporophyte is represented by capsule only. The regressive view is a reverse process of progressive view. Now, what is the argument of the regressive theory? The rationale of regressive theory is that the increasing allocation of sporogenous cells aid in better reproductive ability and is therefore an advanced feature. This argument can be nullified by the fact that evolution of specialized structure for efficient spore dispersal, like peristome in mosses, which is considered as the highest evolved according to the progressive sterilization theory, is a better mechanism for wide spore dispersal and increases more the chance for reproduction. Summary of the theory of regressive or retrogressive evolution. Now, the theory of regressive or retrogressive evolution, that is, the evolution of sporophytes due to the progressive reduction or simplification, is supported by Church 1919, Kashyap 1919, Goebel 1930, and Evans 1939. According to this theory, mature Rickshaw sporophyte of the Hepaticopsida, which is essentially a capsule containing a single layered jacket and a mass of sporogenous cells, is the most evolved. The sporophytes of Bryopsida, showing the highest degree of sterilization and specialization and possessing comparatively less sporogenous tissue, is most primitive. From bryopsida onwards, there is a gradual reduction of sterilized cells and simplification of tissues. The reduction in the sterile tissue of the capsule with simultaneous increase in the amount of sporogenous tissue. Simplification of tissues and loss of specialized tissue structures, like loss of spore dispersal mechanism, that is the peristome, which is present in bryopsida only. Secondly, there is a gradual loss of photosynthetic tissue and loss of stomata. Next, loss of conducting tissue, that is columella. Then, loss of meristematic tissue. Next point, multi-layered capsule reduced to a single-layered one. Next, gradual loss of food and seta. The regressive view is a reverse process of progressive view as discussed above. Conclusion. The rationale of regressive theory is that the increasing allocation of sporogenous cells aid in better reproductive ability and is therefore an advanced feature. This argument has been nullified by some supporters of the progressive theory with argument that evolution of specialized structure for efficient spore dispersal, that is the peristome in mosses, is a better mechanism for wide spore dispersal and increases the chances for reproduction more. Now, remember few things before answering. Number 1, the answer should adhere to the word limit for which the summary is given. 2, write only what you remember well and don't write wrong answers. 3, always underline scientific name of plants, here bryophytes. If you find this video helpful, please like the video, and if you want to watch more of such videos of this channel, please press the subscribe button so that the videos will automatically go to you. Thank you.